Yes, hi everyone. Thank you so much for having me here today. So my name is María. I work at the Faculty of Natural Resource Sciences along with Ottur and Hreiðar that have already given their talks. And I will be joined here today on Zoom by Ingunn Grétarsdóttir that's about to wrap up her master's in industrial biotech. And we're going to give you a brief introduction on some of the microbial work that we've been involved in lately and that directly relates to the need of societies globally to kind of shift into a more alternative and greener ways of chemical synthesis. So the microbes that we're going to be briefly discussing today, they belong to this group or genus that is called Thermophilic, very easy to pronounce. And as you can maybe make out from the name of the genus, thermo refers to the fact that they thrive at elevated temperatures. So they will absolutely not grow below 65 degrees-ish, and they also cannot tolerate molecular oxygen. So if you put them in an environment where there's a lot of oxygen, they will simply die. So we sometimes refer to them as microbial workhorses because they're very biochemically fun. They can do a lot of useful things. And what you will find in the literature when you look for this genus is they are very efficient bioethanol producers. Like they will produce ethanol from a number of substrates and have done so very well, and it's very well documented in the literature. So they're great fermenters, and they're also able to produce other alcohols, not just ethanol, because while ethanol is useful as fuels, there are other alcohols out there that can be used as solvents or as starting materials across industries, and we like to call them simply chemical building blocks. But the production of alcohols isn't a novel idea. We do it industrially on a large scale. We know exactly how to do it. So the question is, if we know how to do it, why bother using live cells? Why bother using cells that don't tolerate oxygen and grow at 65 degrees and above and make things a little bit more complicated? And Ingunn is going to answer the question for why we're bothering. Ingunn?
The current industrial alcohol production methods, there are a few common ones highlighted in the table, as you can see on the slide, but they require higher temperatures, high pressure, and they are fossil fuel based or like use derivatives of fossil fuels, which is, as we know, very wasteful and not sustainable. Also, there are some catalysts that may be used in these processes, which are toxic for the environment. However, we can use living cells, such as microbes, to produce some alcohols. And this biocatalysis requires much lower temperatures, lower pressures, and the best part, no fossil fuels. It also offers opportunities of waste valorization, and they are able to transform like lower value compounds to higher value ones. So of interest now has been the thermophilic carboxylic acid reduction. Try to say that fast 5 times in a row. And this involves the conversion of acids to alcohols using heat-loving bacteria such as Thermophilic.
Thank you, Ingunn. And this is what this biochemical process looks like in a nutshell. So what we do is we feed the cells the acid of interest. And so acids or carboxylic acids, what we mean by that, those are chemicals that have kind of this characteristic functional group that we write as COOH. We feed them an energy source because they need something to grow on, and then we patiently wait. And what we will see with Thermophilic, and you'll also see this with some other bugs, is that with time they will actually convert the acid to its corresponding alcohol. So let's say that our starting acid is butyric acid. With time, what you'll see in the medium in their growth environment, and you'll see a buildup of butanol, for example. So the corresponding alcohol of butyric acid. And this conversion, it doesn't just happen on its own. It's in the hands of these things called enzymes. So these are protein molecules that all living cells have. We have a bunch of enzymes in our body. Basically, every form of life has it, but the types of enzymes are very cell-specific and they will differ between cells, which means that the ability of cells to do this, even within the group of Thermobacter, will differ between different strains because they'll have different enzymes. So what Inggunn did in her master's project, she did what we called substrate mapping, which is why I have a person pointing at a map on the slide. So she basically threw a bunch of acids at different strains of Thermobacter, gave them a bunch of energy, and then she waited. And what we'll see is you'll have different strains can do different things to different acids. And here on the slide, we have highlighted just one Thermophilic strain called AK85. AK stands for Akureyri. It's very original. And the orange columns, they correspond to alcohols that are listed on the x-axis. So the x-axis contains the name of the acids that were used, and then the columns correspond to the alcohols that were produced. So what we'll see for strain AK85, that's by the way an Icelandic strain, it was isolated from an Icelandic geothermal pool, is that it will turn different acids to their corresponding alcohols, but it will do it differently, right? So the first column is the highest ones, and then you'll see some variations. But she did this for a number of strains before moving on, because it's not very impressive to take acids that you get from a foreign chemical company that wasn't necessarily produced sustainably and turn it into an alcohol. So what she did after kind of making the substrate map, she decided to make her own acids using renewable biomass. So we've been collecting algae and drying it. We get some fish meal. We even use lupine, which, by the way, is an invasive species. So if you can use an invasive species for something like this, that's a big plus. And then she used fish waste from aquaculture to kind of start this fermentation process to get these acids. And then when she got the acids, she put them into the cultures of these Thermophilic bugs, waited again, took samples, and analyze them. And the energy source, while she started with using glucose, which you could argue that is kind of a food source and we should be moving away from, she used sugars from algae. So she both managed to kind of make the acid production more sustainable. She managed to do a little bit of waste valorization by using fish waste to kind of make the acids. And then she also tried to use a more sustainable source of energy to begin with. So here you can see on the slide, there's a little pic of what the acid production looked like, and then the little bottles on the end is how we culture the microbes.
So
Ingunn?
yeah, good news. It worked. The Icelandic strain produced ethanol as we suspected it would do, and you can see it on the blue columns. But it also was able to convert some acids in the digestate to other alcohols, as you can see in the orange columns. So our plan worked. We were able to make the alcohol production more sustainable. But however, there is a little bit of a black box. We were originally supposed to use a different method for analysis, but due to instrument failure, we could not. So we tried a novel method. However, it could not tell us which carboxylic acids were present in the digestates before we reduced them. So we just kind of know there were acids. We made alcohols, but we do not know exactly which alcohols were produced. However, we have samples for everything set up and just waiting to be analyzed with this original method, which will hopefully be in the near future. It would be very exciting to see which acids we were able to make via the anaerobic digestion. But that can also tell us more of we might need to tune the anaerobic digestion process. There are a variety of factors which influence both the composition of acids produced and how much we produce. Also, one exciting thing to look forward to in the future is that recently this strain got whole genome sequenced, and we might be able to do some studies to decipher the pathways which are responsible for this reduction. We kind of don't know what exactly is happening. We just know they eat some acids and make some alcohols. Yeah, if you could switch to the next slide, please. Thank you. So in summary, this is basically my process in a nutshell, or the project, sorry. We use waste to make anaerobic digestates, which contain carboxylic acids. Then we can use thermoanorobacters to reduce those acids to their corresponding alcohols. Further downstream processing lets us separate the cells from the alcohols, and we might potentially be able to reuse the cells, which would be great. However, we need to think about like how we want to recover the alcohols from the fermentation broth. There are a few methods we could use, but like one relies on solvents, and then we need to make sure we are using more green solvents, not because they tend to be toxic, and we want to make the whole process more sustainable. So we really need to think about each step of the way. But yeah, alcohols have like a variety of industrial applications. They are used in the pharmaceutical industry, food industry, textile, in the chemical industry, and even in the cosmetic industry. So alcohols are everywhere. So finding ways to produce them in a more sustainable manner would be great, especially in the light of recent geopolitical events.
Yeah, so that's all we have. We will be happy to answer any of the questions that you would have. Feel free to reach to us out via email, and special thanks to Landsvirkjun that partially funded this project. Yes, thanks. Thank you.